Okay, so good afternoon, everyone. We are happy to have Stefan Vasiljevic from Bern, Switzerland, presenting for us today. Stefan is a PhD candidate at the University of Bern, where his research focuses on quantitative and genomic analyses to identify genetic variants influencing hoof and leg traits in Swiss dairy cattle. Stefan holds a master's degree in agricultural sciences from ETH Zurich, with a major in animal science and minors in data science and plant breeding. Stefan gained experience in data analysis through an internship at Swiss Genetics, where he worked on cattle embryo datasets, and he conducted genomic analyses during his master's thesis in collaboration with Qualitas AG, focusing on the genetic architecture of milk urea content in Swiss cattle. In addition, Stefan gained hands-on experience through genetic-related lab work with the molecular plant breeding group at ETH Zurich, as well as solid statistical knowledge through involvement in the teaching of applied statistics. So thank you, Stefan, for being here today and presenting for us. I'll let you take it from here with your presentation.
Thank you very much, Gabi. Then hello, everybody. As Gabi said, I'm Stefan Vasiljevic, a PhD student at the Institute of Genetics at W26 faculty of the University of Bern, that's in Switzerland. My supervisor are Joanna Jacinto and Kurt Droge-Müller, and today I will present you my research on the identification of genetic variants that influence these hoof and leg traits in Swiss dairy cattle. For this work, I collaborated with Qualitas, which is the national organization for the breeding values estimation, and also with the ETH Zurich and INRAE from AgroParisTech for knowledge share and data. I will begin with a background. Body conformation are somehow related to the functional traits, the health traits. One example is poor hoof and leg conformation, which leads to higher risk of hoof-leg disorders. Examples of hoof-leg disorders are hoof lesions or lameness. Lameness are one of the most prevalent challenge in dairy cattle industry, decreasing the welfare and the longevity of the animals and increasing the farm cost. Lameness is really common in Swiss dairy cows, with more than 80% of the cows having at least one cow being lamed. It's also very hard to study these hoof disorders because they are not routinely recorded and they are really hard to quantify. Then the opportunity is to use these hoof legs conformation traits because they are routinely scored to try to study, to reduce the hoof disorders through selection. But for that, we have first to study the genetic architecture of these hoof and leg conformation traits. Then what say the literature about that? The heritability of these hoof and leg conformation traits are about 5 to 37%, 37%, low to moderate. I found also some GWAS studies, mainly additive, and they found several candidate genes, DGAT1, ADIPOR2, FGF23 in Holstein breed, and in a study of Pang and Pausch 2019 in Switzerland, they found 4 associated regions in brown Swiss and 2 main candidate gene RSP04 and BTRC. Now, as I said before, the GWAS, there was only made with additive model, which assume an additive inheritance pattern. I will just show you with a pointer. Just a second, please. There, that's the assumption. That means that the SNV looks like that. The box plot of the SNV looks like that. That means that each addition of the alternative allele increase or decrease the phenotype linearly. But it exists also non-additive models. And they are really useful because then we can detect some rare and large effect variants. The first non-additive model is the dominant one. It is assumed that only one copy of the alternative allele is increasing significantly the phenotype or decreasing the phenotype. And we have also another type of non-additive GWAS. It is the recessive one, which assumes that two copies of the alternative allele increase or decrease significantly the phenotype. What is good or what is the limitation of the GWAS? The first thing is the poor resolution, because if we have a poor resolution in the GWAS, we cannot detect the causal candidate variant or the causal variant. If we have a high resolution GWAS, the problem is that we will find an associated region, but these associated regions are linked, these SNVs in the associated regions are linked through linkage disequilibrium, and then we will get some things like that. A lead SNV, lead SNV with other SNVs that are linked with them. The lead, I just want to mention that the lead SNV is not the causal variant. The causal variant is one SNV that is, it can be the causal variant, but it can also be one of the associated SNVs. Then to try to identify the SNV, we can use the causal variant. We can use conditional and joint analysis, which identify independent signals inside this associated region. And then we can also use fine mapping. One example will be this Bayesian fine mapping, which estimate a posterior probability for each SNV. And then with this posterior probability, we can create a set of plausible candidate variants. This is the example. Then we have the GWAS with the lead SNV there. And with the statistical fine mapping, we get for each SNV posterior probability. And as you can see there, we have a group of variant there that will be in our plausible set of variant. The current knowledge gap in hoof and leg conformation traits. The first one is the missing of genomic resolution. We don't have any high resolution GWAS, and we have also a limited statistical power. I just wanted to show you a study of James and all 2022. He made two GWAS on the same with the same phenotype, but one with a low density SNP and the other one with a very high density SNP data, the imputed SNP data. And as you can see, in the low density SNP data, we don't get any associated signals. But when we use the high density one, we get some hits. For the breed-specific gap, we don't have any study on Swiss Holstein, and we don't have also study on individual trait analysis in brown Swiss. The study that I showed you earlier of Fang and Pausch 2019 was a study was a study on composite trait and not on individual trait analysis. There is also no study considering non-additive models. Then the aim of our analysis, first one, it was to estimate the additive heritability of hoof and leg conformation traits on two main breeds, Swiss Holstein and brown Swiss. Then it was to identify associated genomic regions using additive and non-additive model. Then it was to detect the candidate genes and candidate variants. And at the end, it was to clinically characterize animals carrying the candidate variants. Then for the traits that I analyzed, the first one in Holstein was heel depth, then foot angle, locomotion, bone structure, rear leg rear view, and rear leg set. I analyzed 6 of them. I analyzed all of them, but I have 6 different traits. Heel depth was a measure in millimeters. The minimum value was 10 millimeter and the maximum was 60, as indicated in the red box. The ideal value was indicated in the green box, which was 45 millimeters. The other traits were scored with values between 1 to 9. For BS, I analyzed 4 different traits: rear leg side view, op quality, foot angle, and heel depth, also score with value between 1 to 9. Then I just wanted to mention the genotype and the phenotype data that I used, just to understand a little bit the power of the study. Firstly, the phenotype records are the linear scoring measured by the breeding organization. And it is possible to find in this dataset multiple records per animal and per lactation. At the end, I get a data of 1 million records for Holstein and 1 million record for Brown Swiss. For the number of animal genotyped, they were 87,000 for Holstein and 50,000 for Brown Swiss. Then when I merge both data, the phenotype records and the animal genotypes, I get 21,000 animals with genotype and phenotype for Holstein and 50,000 for Brown Swiss. The ETH shared with me the whole genome sequence level imputed SNV panel for each one of these animals, with more or less 37 millions of SNVs in the data for Holstein and 26 millions SNV for Brown Swiss. I also made a subset of these imputed SNV panel, a high density SNV panel of about 700,000 SNVs for Holstein and the same for Brown Swiss. For the estimation of the additive heritability, I use the high density SNV panel. I also use GCTA for the variance component analysis. I use the first 4 principal components as fixed effect, the additive genomic relationship matrix as random effect, and the corrected phenotype. What does corrected phenotype means? I used a mixed linear model to correct the raw phenotype. I corrected it for classifier, age, days in milk, time of day, and farm being the random intercept. And as you know, in each of these mixed linear model, you get errors, the residuals. They are there. And these residuals contains the genetic variation of the animals. And I call them corrected phenotypes. And I use only corrected phenotype for the subsequent analysis. Then the results for the estimation of the heritability, I found low to moderate heritability in line with what I found in the literature. In Holstein, the highest value was for bone structure, about 28%, and the lowest one was for locomotion, about 10%. For Brown Swiss, the highest one was for all quality, 26%, and the lowest one was for heel depth, 10%. What is important to say is that we get, for similar traits, we get similar heritability between both breeds. That means that between bone structure and hog quality, we get more or less the same value, about 26 to 28%. What's also important to say is that in Holstein, we get a little bit higher heritability. Now, I did my first GWAS using this high density SNV panel, about 700,000 markers. I made it with GCTA, and this is the model of GCTA for the mixed model. And normally, this Xi SNV is the vector of genotype for the testes and SNVs, and they are coded 0, 1, 2, like that. 0 for the homozygous reference, 1 for the heterozygous, and 2 for the homozygous alternative. For the non-additive GWAS, I just recoded these SNVs. For the recessive, I recoded 001, and for the dominant 011. I also use a threshold of 1 multiplied by 10 minus 6 for the suggestive QTL. Then I just want to show you one example of the GWAS that I made. This is for O quality in brown Swiss. The inner circle is the additive model. The middle one is the dominant model, and the last one is the recessive model. The gray line is the suggestive threshold of 1 multiplied by 10 power of minus 6. And I also defined a significant threshold, which is the black line. The threshold is about 5 multiplied by 10 power of minus 8. Now I want to show you a second example, which is bone structure in Holstein, and this time I will show you one more time the additive, the dominant, and the recessive model with the both threshold. Each time that SNV, that the marker is above this suggestive threshold, it will be colored in pink. And as you can see there, for example, I get hits on the chromosome 5 for the dominant and for the additive model. I just want to mention something now, which will be very important for the rest of the presentation. Sometimes you get overlapped regions between the models. I just want to say that now, and I will explain that a little bit later more. Then for the results, the overview of the results are in this table. I get 45 suggestive QTL with this method across traits and breeds. I get 22 additive QTL, suggestive additive QTL, 16 suggestive additive QTL with the dominant model, and 7 with the recessive model. Now, once that I found these suggestive QTL, I made a second GWAS only on chromosomes that harbor these suggestive regions. And one more time, I use GCTA. And I use the whole imputed SNV panel for these chromosomes. I show you before also the threshold that I use to define the significant QTL, 5 multiplied by 10 power of minus 8. I will show you an example. I think it's good later. This is the GWAS with the high density panel, about 700,000 SNVs that I showed you earlier. Now, if we focus only on one of these regions, one of these QTL, on chromosome 22, for example, you can see that I get a hit, that I get a significant QTL. For this chromosome, I will use all imputed SNVs and make and use the mixed model to get the p-value of each SNV. And as you can see there, I found a very good associated region with the trait. Now, another example will be for O quality in broad Swiss on this chromosome 25. You see that markers are above the suggestive threshold, but lower than the significant threshold. When I use whole imputed SNPs for the GWAS, I found an hit on the significant level. Now I want to show you another example. It's on the chromosome 7. One more time, one marker is above the suggestive threshold, but lower than the significant threshold. When I use whole imputed SNPs, I didn't get any hit on the significant threshold. At the end, I get 25 significant QTL, 13 with the additive model, 10 with the dominant model, and 2 with the recessive model across traits and breed. As I said before, sometimes I have hits, I have associated regions in several models. There we have the same regions found in the additive and dominant model on the chromosome 18. And each time that I found that, I only took the additive QTL and not the dominant one. Why? Because firstly, normally the signal is stronger in the additive model, and because the additive model explained the most of the variation. At the end, I get 15 single significant QTL, 13 with the additive model, 1 with the dominant model, and 1 other with the recessive model. The QTL found with the recessive model is in bond structure for Holstein, and the dominant one is for high quality in brown Swiss. Now that I found the significant QTL, I fine map this QTL. I try to find the causal variant. And this is my strategy for fine mapping. Firstly, I defined the independent association signals using conditional and joint analysis with GCTA. With that, I identified the lead SNVs, and then I extract around each lead SNVs 2 megabase windows, 1 megabase downstream and 1 megabase upstream. This is one more time the example of the GWAS for bond structure in chromosome 22. I found only 1 independent signals with 1 lead SNV. I extract 2 megabase windows around this lead SNV, and I get this zoom plot. Once that I get a zoom plot, I get the 2 megabase windows, I use the Bayesian fine mapping tool called Painter. What is good with this Painter is that I can use priors. And as priors, I use as priors the SNP-F functional impact. You can find modifier, low, moderate, and high. Moderate and high being the impact of protein coding variants. And I also use this phase score that are a combination of evolutionary importance and heritability, which is calculated based on 34 traits. You can find more information about this phase score in the paper of Chang and Hall 2019. With this Painter, I calculate for each SNV in these windows a posterior probability, and then I define the 95% credible set. This is one more time the example of before with bond structure, the 2 megabase windows, and we can see there that Painter found 1 causal variance for bound structure on chromosome 22. This is the list of the candidate genes for the 15 QTLs. If you want to know more about that, you can ask me later. And this is the 17 candidate variants found by Painter. I found 3 candidate variants in Holstein, 1 with the recessive model in bond structure, 2 other ones in locomotion with the additive model. I found also 6 candidate variants in yield depth in brown Swiss with the additive model, 8 others, 1 candidate variants in oak quality in brown Swiss with the additive model, and 1 candidate variants with the dominant model in oak quality for brown Swiss. You can see also some genes that are marked that are highlighted in bold, and this is the genes. This is the variants where this is the protein coding variants and the associated genes. I found 4 different protein coding variants on YAL1, GCC2, RIPOR1, and FGF8. Then this is one more time an example of additive candidate variants for O quality in brown Swiss. You can see there that Painter found only one candidate variant for this QTL on RIPOR1. If we examine the distribution of the corrected phenotype across the genotype, we can see there a good additive inheritance pattern, as expected. Now, this is another example for the additive candidate variants for O quality in brown Swiss, but in chromosome 26. And you can see there that Painter didn't find only one candidate variant, but find 4 of them. For the recessive candidate variants in bone structure, I already said that Painter find only one candidate variants on YAL1. And this is the distribution of the corrected phenotype by genotype for this variant. And you can see that these variants follow a recessive inheritance pattern with an effect size of about -1.6, which is huge because it's more than one standard deviation for the corrected phenotype, which means that the animals that are carrying 2 times the alternative allele normally should have bigger joints. And we went on farm, we found 7 Holstein females in farm, and we did an examination of these females. We found 3 heifers and 4 cows. For the clinical findings, we found, as expected, that these animals have very large joints, and one of these animals was lamed. It was the oldest one, actually. You can also see there that the joints are big, but also that you can find this strange structure on the joints there. And we did an ultrasonography on these joints, and we find on this structure, and we found liquid inside. Joanna, that is a very good vet, said me that is synovitis, which means an inflammation of the synovial membrane, and with effusion, that means with liquid inside the joints. What is the function of this YAL1? This YAL1 produces hyaluronidase 1, an enzyme that breaks down the hyaluronic acid. The mutation of this YAL1 causes mucopolysaccharidosis type 9 in humans, which is characterized by soft tissue, joint swelling, cartilage abnormalities, and reduced bone quality. This is an MRI of one patient with this disease, and you can see liquid inside the joints there. The predict effect of these variants on cattle, as I said, this variant is a stop-gain variant, and it may disrupt the hyaluronic acid breakdown, leading to weaker cartilage, altered hoof bone structure, and accumulation of hyaluronic acid. The limitation and the perspective of my analysis. The first thing is that I use subjective phenotypes. It's phenotypes that were scored by classifiers, and that was done subjectively. I tried to account that in the model, but it's never 100% correct. Then I use additive dominant recessive GWAS separately, which increase the number of statistical tests, and which means also that I should also increase the threshold, the threshold of my GWAS. One more thing is that you can find also some models that take into account the additive and the non-additive model in the same model. Then the fine mapping windows used, I use 2 megabase windows. Actually, I could use 5, 10, 20 because cattle has long range LD. Then the priors used in Painter, I used this phase score that covers 17 million variants, but actually in my dataset I had much more variants. 36 millions for Holstein and 26 millions for Brown Swiss, which means that some variants didn't have any phase score, which is a bias for pi-inter, and that means to bias results. That leads to bias results. And I also didn't have any functional validation. That means that I didn't have any expression data to find EQTL. The take-off message is that we made the first high-resolution analysis for these hoof and leg conformation traits. The traits are polygenic, driven by many small effect loci, and the non-additive GWAS models reveal important large effect recessive variants such as YAL1. These non-additive GWAS were really important because with them we can monitor deleterious allele breeding population and then improve the future genomic evaluation. Now I just want to thank you, all the organization people that helped me. First one, it was La Confédération Suisse, ASR, and the Schaumann Stiftung for the financial support, Qualitas and ETH, as well as INRAE for the knowledge, and the breeding values and the breeding organizations for the data. I just want to mention that I submitted a paper about that in January. And in this paper, you will find much more results on YAL1, on these recessive YAL1 nonsense variants, which has also an effect on the longevity of the Holstein cattle, but also on the other conformation traits. Do you have any questions?
Thank you so much, Stefan. That was a great presentation. I will open the floor up for any questions in person or online. If you want to raise your hand and just let me know. I'll get us started. I was just wondering if there's anything in place for the future to maybe have genetic testing or something after seeing this association with the different variants and the negative phenotypic score or phenotype or score.
Not now, no. I didn't present the results to the breeding organizations, and they don't know actually the results now. I will do that normally next week.
Okay.
Or in 2 weeks, then no, nothing is done now.
Okay, sure. Flavio? I can't hear you on my end, Flavio, if you were talking.
Can you hear me now?
Yes, yes.
Yeah, is okay now?
It's a bit quiet, but I think it's okay.
Yeah.
Okay. Okay, so I will try to get closer to the camera. So, Stefan, great presentation. Thank you for doing that. I have a question for you about the decision that you made for the models of the GWAS. And not the additive model, that's the standard one to do, but you decided for the dominant and recessive model.
Yes.
But when you do that, you are assuming that there is complete dominance. Either it will be recessive or be dominant. In other words, the heterozygotes are going to behave as one of the homozygotes. So it's complete dominance. So,
I have difficulties to hear you. I'm really sorry, and to understand you.
Okay.
Can you write it perhaps, and then I can.
Let me. Can you hear now better?
Yes.
Can you hear now better?
Yes, it's better. Thank you.
Okay. So I will repeat. Now, my question was about the decision of your models.
Yep.
And so the additive model is a normal, common model because we have interest in the additive effect for later on for sake of selection. But then you decide that the dominant in the recessive model.
Yes.
But when you do that, you are so you are assuming that there is complete dominance of either of the two alleles, right? So the heterozygote is going to be the same as the one of the homozygous.
Yes.
But how about if you have incomplete dominance? So because you are losing the ability to map the incomplete dominance.
Mm.
And the complete dominance is just a particular case of the incomplete dominance. So I wonder why you didn't fit the model where you fit the genotype as a classification variable. So 3 classes, right?
Yep.
So the genotypic effect. And from the genotypic effect, you can estimate the additive effect and the dominance effect of the genes. Or even using a regression model where you fit 2 regressions, one to estimate the additive effect, that will be the normal recoding of the alleles, let's say -1, 0, and 1, or 1, 2, 0, 1, 2. But then you regress on 0, 0, 1. 1 for the heterozygous, 0 for the homozygous. If you do this simultaneously, you estimate the additive action of the gene and the dominance action.
Yes.
Right? So
Yes.
I don't know. I think that would be quite informative in your study too, because you would capture this dominance that's not complete.
Yep.
I don't know if you thought about this or
Yeah, I thought, yeah, that was the limitation. I say that was the limitation of my work. It was that I didn't model both additive and non-additive model in one linear model. But
okay,
yes,
so I didn't get what you are
yes,
trying to say in that sentence. Yeah, I think because that would be you could capture more genetic variation. Maybe you would have more regions to look at. That you miss it by just using it is full recessive or full dominant, right? Full dominant.
That's
But great work. I'm looking forward to read your paper. Very nice, very interesting. Thank you so much, Stefan.
Thank you very much. I will try to improve that for the next paper. I'm a beginner. And then I also learn with the time. And when I finish the results, when I finish the analysis, I just thought about that, and yeah, it was a little bit too late. And for the next one, thank you very much, Flavio.
Thank you. There is a question in the room here from Julia.
Hello, Stefan. Great presentation. I was wondering if you have compared Holstein and Brown Swiss and have identified any common candidate variants between them. And also, did you compare the phenotypes between breeds? And did one tend to have better scoring than other?
No, you cannot compare the phenotypes between the breeds because they are different traits, firstly, and they are differently measured. They are differently scored too. And that's the first thing. The second thing is, I didn't say that, but in my population, I had several populations. The first one was the Holstein population. You found several breeds. The first one is the Simmental, the second one is Swiss Fleckvieh, and the third one is Holstein. I'm now analyzing that, trying to find some difference or some similarity between these breeds. But then when I compare the Brown Swiss and the Holstein, it's 2 different breeds, and I think it's not really a good thing to mix them up or to make a meta-analysis with these 2 breeds. They are completely genetic different. Yes, it is. Did I answer the question?
Yes, you have answered that. Thank you so much.
Thank you. Are there any other questions for Stefan? Oh, okay. I think your presentation was very clear. Thank you, Stefan. We did end a little bit early, but I think we had a great discussion, so we can end here, I think. So thank you for joining us again. It was great. We'll be posting this recording to YouTube next week, so I'll be in contact with you regarding that. But thank you everybody for coming in person and online. We will see you next week.
Thank you all. Thank you. Ciao, Gabi.